So I think we're going to have to move on to our final speaker, Andrej Sali, who is at RCSB PDB and University of California, San Francisco. And he's going to be talking about from integrative structural biology to cell biology. Andrej.
Hi, good morning, everyone. I'm very grateful for this opportunity, in particular because the Protein Data Bank has been so enabling of our research in so many ways over so many years. So thank you. I would like to focus on model integration as opposed to data integration. And an example of data integration would be using cryo-EM maps and chemical crosslinks to get a molecular model of the system. An example of model integration would be combining structural models from structural biology with other types of models, such as molecular network models, say from systems biology. And I think for maximizing the impact of structural biology on cell biology, model integration in particular will be very, very important. And so I'd like to talk about that today, about our early efforts on this topic and highlight this topic to you. Of course, this reflects the research interest of our group at UCSF, where we aim to develop, apply in collaboration with others and disseminate, importantly, various modeling methods for describing biological systems to facilitate biology, but also biomedical discovery. And the basis of our approach, this is going to provide the starting point for the main topic, is integrative modeling, which we define as that type of modeling that in principle uses all available information about the system of interest. And this information can come from experiments on the system of interest or from prior models. And these prior models may include physical theories, in the case of structural biology or molecular mechanics force field, for example, statistical inferences, and other prior models, such as subunit structures when you're interested in a complex. Of course, by using all information, at least in principle, one maximizes accuracy, precision, and completeness of resulting models, and that's the motivation for doing integrative modeling. The way this works in a simple sentence, you can say that you express the problem as an optimization problem in which you find all models for which computed data match the experimental data within some acceptable threshold. And we do like to think about this problem in an increasingly Bayesian fashion, but I won't go into the details about that here. So information in this approach, importantly, can be used in many different, in 5 different ways: to represent the components that are being assembled into a complete model, to specify spatial restraints that define the arrangement between the components, to perhaps inform the sampling, to filter the models, and only to assess the models. We have been implementing our efforts in this area on the computational side in an open source software program we call IMP, which we like to use in our applications, but we also distribute it, and we are very happy when other people use it for their applications and for their method development. And over the years, we've applied this approach, this software, to many specific systems, always in collaboration with experimentalists, to challenge ourselves and hopefully maximize our impact. There is a community aspect to all of this, which is, I think, very important, especially for integrative. It's always important in science, but even doubly so for integrative approaches. So I'd like to make some comments on this topic, also because it's very much involving the Protein Data Bank. For a number of years now, there were groups of people working with the Worldwide Protein Data Bank group, with participation of various Worldwide Protein Data Bank groups, to construct an archive for integrative structures and the data on which these structures are based. This archive is now very much alive, and people can deposit their structures and data into it. There is some advantages that this archive offers. You can use representations other than atomic representation of the standard PDB, such as coarse-grained representations. You can represent processes, multiple states, and as I mentioned, you can incorporate data that are not traditionally part of the PDB into this particular archive. And in due order, this will merge with the PDB, I believe, so there will be one unified archive for everything, including integrative structures. I acknowledge people, important people, some of them in this effort below, but I'll also do that at the end. A particularly interesting aspect of this resource is its federated nature. In fact, there's multiple databases that contain different kinds of data that are bidirectionally linked with the resulting models. And an important aspect is that these individual databases are often created not by the PDB, but by the community that's generating the data, where there is all the expertise and motivation to take proper care of their data, but at the same time connected with structural biology, thereby expanding the scope of structural biology, the impact of structural biology, I think. So let me now, after this quick outline, move to the main topic that I'd like to make comments on, and that is the integration of models as opposed to integration of data into a model, integration of different models into types of models into a bigger model. And that, as you can imagine, I hope you can imagine, or I submit, is going to be very important in all likelihood for cell mapping, for obtaining some kind of a complete and useful model of the entire cell. I will discuss our recent publications noted on this slide in case you wish to read more about it. The context for much of, though not all our efforts, our group's efforts in this area is provided by the Pancreatic Beta Cell Consortium, which is an open source consortium of maybe 15 or so research groups at this point, including experimental and computational groups led by Ray Stevens, covering a lot of different types of expertise in experimental and computational methods, as well as artists and engineers and so on, coming all together in an effort to come up with a framework for mapping a cell and apply that framework to the pancreatic beta cell in particular. There are, of course, already existing approaches to wholesale modeling. There's a number of them. I'll just give you a sense by listing three real quick. Marcus Covert at Stanford and colleagues are emphasizing network types of models, differential equation types of models for describing a cell comprehensively in a very impressive work. Then Lucy Schulten's group is a little bit more spatiotemporal. She's using as a core representation a grid, a spatial grid covering the cell, with every grid point having the ability to undergo some reaction or some process, thereby conveying some spatial aspects of course, of the cell. And then there's other approaches that are maybe more emphasis on the visualization side, such as CellPack and related by Art Olson, David Goodsall, Graham Johnson, and others, where there's very advanced visualization tools applied to reflect what is known about cells and to some degree model them in the process as well. And there's many others. But I don't think that any one of the existing approaches is quite ready to, or convincingly ready to be applicable successfully to a demanding model of the cell. And we have speculated about various aspects of what such a model should include, and there are some of them listed on this slide. I can't go through this in any detail given the time, but what I'd like to point out is that a model of the cell in the end will probably have to be multi-scale. In other words, it'll have to represent very different kinds of aspects of the cell, relying on different representations. There's reactions, there's structural organization, there's spatial-temporal organization, there's molecular networks, there's abstract logical relationships between components and within processes. And for a model to be both informable by the information available as well as useful for our understanding of the cell, it'll have to contain multiple presentations of the cells, in other words, be multi-scale. And very importantly, these different representations will have to be in harmony with each other. They'll have to be consistent with each other. What one type of model is saying will have to be consistent with what other type of a model is saying, without defining what harmony means exactly, but I think we will have some sense about what it means. And that's non-trivial. That is, to achieve this requires an integration of different model types to get a bigger model. And that's the problem we're addressing here, because we believe it's important for cell mapping and for maximizing the impact of models of the kind that are generated by structural biology in the field of cell biology. And so here's a very simple idea. We want to achieve this through some kind of a divide and conquer process, where we take as input any existing model constructed by anyone, by any existing method, about any part of the cell, any aspect of any part of the cell. These are models that science generated about the cell. And then, however it got them, doesn't even matter. And then we want to take these models together and harmonize them with each other by a process we call metamodeling. And just to define this even more, describe this even more clearly, what the problem is, I contrasted to integrative modeling, which I briefly already described at the beginning, where you take different types of information to get a model that's consistent with input information about the system of interest. Here, you're taking different types of models and put them together into a self-consistent picture of a bigger system. A very special case of metamodeling is, of course, when you take, say, two subunit structures in a complex and through molecular docking construct a model of a complex. You have used two separate models to get a bigger model, making the subunit models, if you model induced fit, more self-consistent with each other in the process, hopefully improving the accuracy of the complex model. The problem is when you want to do that across different types of models, and that's what a non-trivial aspect of combining different models with each other. That's what we're going to address here. And as a test-bed system that we used, we selected the glucose-stimulated insulin secretion in pancreatic beta cells. I mentioned the pancreatic beta cell consortium. That system has been described at many levels in the organization in the biological hierarchy. There's organs that participate, such as liver and pancreas. There's cells or groups of cells even. And of course, there's pathways and individual molecules inside the cells as illustrated schematically on this picture. And we want to put it all together and make it self-consistent. And so we will now slowly become more specific in terms of describing what we actually did so far, and a little bit also what we haven't yet done. We have so far in this proof of principle 8 input models. They are schematically illustrated in this circle here on the right. I'll just go real quick through them, and I'll tell you about each one a little bit more later, so you get even more sense. But going down slowly in detail, there's a model of how liver interacts with the pancreas, a very simple linear ordinary differential equations type of a model. There's a model of the organs themselves, just consisting of a number of cells, each one of them. There's a increasingly higher resolution model here of vesicle exocytosis, which is provided by a coarse-grained Brownian dynamics simulation of trajectories that vesicles undergo in the cytoplasm as they approach the cell membrane to exocytose insulin. There's a molecular network model of insulin signaling There's some data tables, two different kinds of data tables that quantify how insulin concentration changes as a function of time and glucose concentration in the stimulus. There's some additional molecular network, ordinary differential equation types of metabolic pathway models involving various components of the system. And there's also a very molecular structural biology type of a model, namely a virtual screening of a library of small compounds against the glucagon receptor, GPCR, sitting in the membrane here. And very different models representing very different scales. Each model is formally defined, and we want to put them all together and update each model in view of all the other models, so that we can, in some sense, say we have a self-consistent set of updated models at the end of all this process, to which then collectively will provide a better, hopefully more accurate, more complete, more precise picture of the entire system. And mind you, this is what scientists do all the time in their heads. Of course, informally. All we're trying to do is do this as formally as we can, in the hope that we can then do it on a large scale, automatically, objectively, and maybe even more accurately than even the best scientists can do in general. And so here is the algorithm. So that was still the goal, and now here is the actual algorithm for implementing these ideas. So we have 3 steps from the input models. We go one by one, and we convert each model into a common representation. In particular, we use a probabilistic description. I'll show you what it is for each model. But because each model is expressed in the same way, then in the second step of coupling, we are in a good position, or a better position than before, to couple each model to at least one other model in the input set. And coupling is just imposing another type of a model that relates a pair of input models converted into their surrogate models. And then after we have couplers, as mentioned, then we can use backpropagation from statistics to update the values of the variables in each of the surrogate models based on the relationship between this model and other models through the couplers. Of course, taking into account the relative uncertainties of all of these variables whose distributions we are updating as is commonly done standard fashion in backpropagation. And at the end of this, we have a set of updated surrogate models, which can then be used to update, trivially so, the input models as well. And that's our meta model, as we call it, a set of refined, harmonized, coupled, self-consistent, one hopes, input models. And so to give you a sense, and this is now going quick, but just to give you a sense of formally and more specifically what the input models are, here they are. 2 of them are just tables here in the middle. Some of them are systems of ordinary differential equations. Some of them are trajectories of coarse-grained Brownian dynamic simulations. Some of them are molecular docking lists. Some of them are metabolic metabolism related sets of variables coupled with each other on the lower right. So these are the models, and then they're converted, as I mentioned, into this common probabilistic language by using the formalism of probabilistic graphical models, Bayesian networks, which is just a way, a general way developed by computer scientists to describe joint distributions of random variables. You can visualize them with directed networks like this one here, but it's a language that you can use across any kind of a model to encode that model. And once that is done, and specifically for these 8 input models, the probabilistic graphical models that we derived are shown on this slide, then we can start defining coupling variables. Sometimes this is common variables, like insulin concentration may occur in different models, but it's the same variable, and it provides the coupling between these different models. Sometimes the coupling is a little more indirect, may require an invented third variable, and sometimes it's actually quite guessed. You're not that sure about it, but you can guess maybe what it could be. And you can propose and see what the consequences of that guess are using this mechanism I'm describing. So while we haven't, and that's a disclaimer, obtained coupling couplers for all sorts of models and so on, that's a super difficult problem. Actually, the core of the problem I'm talking about, I think we are providing a little bit of a framework to think about how coupling might be done more generally than has been possible so far for across a more heterogeneous set of models, I guess I'll say. And then you do the backpropagation using the standard rules to get what we call a metamodel. And so this process has a number of advantages that I can't, again, discuss in any great detail, but I'll just highlight one or two here. For example, it clearly divides and conquers not just the construction of a bigger model based on smaller input models, but it divides and conquers the expertise as well, because the input models can be generated by different modelers or different scientists who are experts in their fields. So it facilitates collaboration and sharing of resources that way. It also divides and conquers computational effort required, because you can separately compute each individual model on different kinds of computers in different points on the planet, different locations of the planet, and then put it all together. And it has a number of other advantages that listed here briefly, but I'm not really discussing them. And one important advantage, though, is that there is an ability, at least sometimes, or at least in principle, when things work out, or at least there's a promise of this contextualization, of having information flow from between variables from different models that may be of different types and representing different scales of the entire system that's being described here. And obviously that's very important. It's exactly these kinds of insights that one wishes to obtain when you're combining different models with each other, usually in your head as a scientist. But
Andre, there's 2 minutes, okay?
Yep. Here you can do it formally, at least sometimes, and that's illustrated by this example of where what's happening at the molecular atomic level with the GLP-1R receptor, and what's happening at the organ level with glucose, insulin, concentration, interdependence. This can come together, of course, only when you consider both models together, and non-trivial relationships between these variables emerge that were totally hidden, non-existent when you consider separate models in isolation from each other. And so all this then leads to another idea inspired to a large degree by the protein data bank, but now expanded to cover, at least aspirationally, cell biology, not just structural biology. Imagine a protein data bank that's expanded. This is a virtual thought. It's a Gedanken experiment. Imagine a protein data bank where you can deposit not just the structures, but any kind of a model. And imagine not just a static database that contains all these different kinds of models, but a resource that in addition has a metamodeling type of an engine, such that when new models or new data are deposited, there is an automated update of all the other models that are coupled with the new models or new data to get a better meta model in the output. And if this can be done well, lots of technical problems, lots of scientific problems are still remaining in this. But if this can be done, then that one could imagine would be a good framework for the scientific community, not any individual or individual research group, to eventually map the cell in a good way. And then this is the end of my presentation. I highlighted integrative modeling. I actually forgot to say that it's very much enabled integrative structure modeling of complexes is very much enabled by the component structures that are already available in the protein data bank, including those component structures that are computed now by efforts like AlphaFold and RosettaFold and others. And in turn, these structure models can then be also disseminated through the worldwide, should be disseminated through the worldwide protein data bank. And then I emphasize the idea of combining models, the need for combining models as opposed to data to get a model, and describe one possible approach, early days of one possible approach to address this problem. And especially in integrative biology, one always, of course, depends on lots of contributors. I'd like to highlight Barak Ravich, a key postdoc in our group, who's now a group leader at Hebrew University, who was absolutely instrumental in this effort, as well as Liping Sun, who is at the iHuman Institute at Shanghai Tech, let's say, associated group, who was also extremely key in all of this, and Kate White, who is a key person in the Beta Cell Consortium, keeping us honest, and on the biology side, very much so. And our contributions to the Worldwide Protein Data Bank have been very much both inspired as well as enabled by Helen Berman, but also Stephen Burley, who's very supportive in so many ways of what we're doing with the integrative methods in the context of the Worldwide Protein Data Bank. And there's many others, but that's it. Thank you so much for your attention.
Thank you, Andre. Thank you very much for a very exciting talk. If there are questions now, we would like to hear them. Does anyone have anything to ask of Andre? Okay, so Andre, I think in some ways, a lot of what you have said, we're probably going to talk about in the broader discussion that we're about to have in a few minutes, really having to do with the, where are the boundaries? Are there boundaries in terms of all these different databases, and how do we enable biology? What's the best way to do it? The PDB has been a great example of greatly successful, and then going forward, how we're going to help biology go to the next place, where we're rapidly in the next place. We're going to have to be very clear and careful about how we do this, so we are able to be as effective as possible. Does anyone have any comments about that, or should we? Jane has a comment or question.
A much more limited and more provincial question, really. You talked about handling uncertainties, and Bayesian systems are very good about propagating uncertainties, but not necessarily determining them in the first place. And I think for one big part of this, which is all the existing PDB structures, the structural biology community is still struggling with, and maybe hasn't even started to struggle as much as it should, with finding out and representing uncertainties. And there are some interesting pushes in that direction. Of course, the big difficult point about it, which you must have thought about, is telling experimental or calculational uncertainty, or for instance, really different models, not just local uncertainty in the well that you're in, versus real mobility. And do you have any thoughts or experience with that in this larger context, with things that might help for the structures?
I think you made an excellent comment, and a very deep one as well. And I completely agree. There is, I believe there's a need for Bayesian approaches, unified Bayesian approaches to think about uncertainty in any kind of a structure model that's deposited in the PDB, not all of them, including traditional structural biology models as well as integrative structure models. There is some discussion and hopefully soon some real effort to do this properly at a community level, let's just say. There is going to be very soon, I can say that now, a pipeline for assessing integrative structure depositions as part of the deposition, applied retroactively to the entire contents of the integrative PDB archive as well. Like you said, there are many different kinds of uncertainty. There's not just, and Bayesian approaches are best for propagating it, not for defining the input uncertainties. But you, even on that front, you certainly are in a better position by applying Bayesian reasoning. At least you know exactly what you're doing and what the assumptions and approximations are. Yes, we are thinking, if that's what you meant, about using multiple types of models to model the same system. In some ways, structural biologists do that already when they are applying a single state or multi-state or high resolution or low resolution representations of the same system. You don't know which one is the best one, and maybe you do a model selection type of approach, maybe you do model averaging type of approach in the future a little bit more commonly than is currently the case in order to obviate some of these problems and subjectivity. So there's just lots and lots to say on this topic, a very key topic, because obviously quantifying the uncertainty of any kind of model is key to its proper interpretation, which in the end is what we are all after.
Yeah, that's definitely key.
Helen, may I jump in and just ask? I think we've started the panel discussion.
Yeah.
So by definition, and I wondered if Chris Sander would be able to jump in at this point.